As we conclude our discussion on the structures of the brain, we'll now examine some of the structures that surround, cover, and protect the brain, in addition to some of the chambers that are found within the brain. So we'll start with the coverings of the brain, which are collectively referred to as the meninges. And in humans, we see that the meninges surround the brain and surround the spinal cord. And in both structures, they have three layers. They have an outer superficial layer that's referred to as the dura mater or the dura mater. And its function, at least in the brain, is to anchor the brain to the inner surface of the skull. The middle layer is referred to as the arachnoid mater or the arachnoid mater, and it has small little extensions called arachnoid granulations. That is, they're these little extensions of the arachnoid mater up into the dura mater, or that project up into the dura mater, and some of these projections will project up into large cavities, which collectively function as veins, which I'll show in this diagram here. So we see in this diagram, we see the bones of the skull. Immediately underneath that, we see the layers of the dura mater or the dura mater in gray. In humans, the dura mater has two layers. It has a layer that's right underneath the bone, which is referred to as the periosteal layer of the dura mater, and then the deeper layer is referred to as the meningeal layer. In the space between the two layers, we often see large cavities which act as veins. In this case, there's a large vein in the center of the brain that's referred to as the superior sagittal sinus, which serves to collect blood from the arteries and capillaries that are nourishing the brain and eventually deliver it back to the heart. In this diagram, we also see the arachnoid mater here in kind of a yellowish orange color, and we see little projections of the arachnoid mater called the arachnoid granulations or the arachnoid villi, which project from the arachnoid layer up through the dura mater and into the cavities like the superior sagittal sinus. What these will do is they will help function to drain cerebrospinal fluid, which is found underneath the arachnoid mater, and that will help the cerebrospinal fluid drain up into the superior sagittal sinus and back towards the that fluid will then be able to be drained back towards the heart. The deepest layer, or the deepest of the meningeal layers of the brain, is referred to as the pia mater or the pia mater, and it helps to anchor small blood vessels, capillaries, things like that, to the surface of the brain. That's a very delicate layer that rests on the surface of the brain, and when you're doing dissections in a lab, you can often see this layer as kind of very transparent, but it's very shiny. It almost looks like a very delicate layer of plastic wrapping around the brain. So here it is here, the pia mater or the pia mater resting directly on the gray matter of the cerebrum. As I mentioned earlier, there's a space that's located between the arachnoid mater and the pia mater, so that it's underneath the arachnoid mater but above the pia mater. That's referred to as the subarachnoid space, and typically it contains a thin layer of fluid that's referred to as cerebrospinal fluid, which is abbreviated CSF here. Cerebrospinal fluid functions to cushion and physically support the brain because the brain is mostly made out of neurons and blood vessels, and so it doesn't really have a lot of structure to it. And if there were no fluid surrounding it, the weight of the brain and the blood vessels that are around it would actually cause the brain to kind of crush itself under its own weight. So the cerebrospinal fluid helps to essentially kind of suspend the brain within the subarachnoid space. It does function partially to nourish the brain as well, although this is mostly, nourishing the brain is mostly the function of the capillaries that are embedded within it and on its surface. But the cerebrospinal fluid does function to nourish the brain because it does consist of a lot of things that we see in the bloodstream. As we see in a minute, it comes from the blood. Cerebrospinal fluid is produced by capillaries of the bloodstream. So in that regard, cerebrospinal fluid contains water, electrolytes, as well as oxygen dissolved in it. But it does not contain any other substances within it, because we saw that the blood-brain barrier prohibits a lot of things that are in the bloodstream to leave the bloodstream and enter the brain tissue, simply because of how sensitive the brain tissue and the neurons are to any changes in the chemistry that surrounds them, and the chemistry of the fluid that bathes the brain. So we'll see that cerebrospinal fluid drains from the subarachnoid space into the durosinuses, and the durosinuses, which are the layers that exist between the two layers, the dura mater, are very similar to large veins, and they do permit that cerebrospinal fluid to return back to the blood, and then that blood that's in those durosinuses will eventually be drained back towards the heart. So here once again we see the arachnoid granulations, or the arachnoid villi, which project from the arachnoid mater up through the dura mater and into the superior sagittal sinus, and that allows the cerebrospinal fluid to drain from the subarachnoid space that is there. So now that we've examined the connective tissue coverings that surround the brain, we can take a moment to look at the chambers within the brain. And collectively, these chambers are referred to as the ventricles of the brain. And these ventricles are open spaces within the brain where cerebrospinal fluid is formed from small capillary clusters that are in some of these ventricles, and cerebrospinal fluid also circulates through these. So there are, within the cerebrum, there are a right and a left lateral ventricle, one within each of the cerebra, one within each of the lobes of the cerebrum, that contain small capillary clusters called choroid plexuses, and these choroid plexus structures are the capillary clusters that produce cerebrospinal fluid by filtration. So what happens is blood pressure in those capillaries will force some of the fluid out of the blood, but not any of the cells or a lot. It will also not allow any of the materials or a lot of the materials that are in the blood and plasma to enter the ventricles of the brain, but fluid and some dissolved electrolytes and some nutrients like glucose and oxygen will be found in the cerebrospinal fluid. But the filtration process is very selective in those capillaries, and it doesn't really allow much material to leave the capillaries except for some water and a few nutrients. So examining those structures there, we can take a look at a diagram. Here we see in this diagram from the textbook that there is, in this viewpoint anyway, we've sectioned mid-sagittally through the brain. So we're looking at essentially the right cerebral hemisphere here. This is kind of an outline of where the right lateral ventricle would be in the right cerebral hemisphere. There would be a similar one located in the left cerebral hemisphere, which we can't see in this diagram because it's been removed. But we do see the presence of choroid plexuses in there, which are producing that cerebrospinal fluid by filtration. From the lateral ventricles, those two lateral ventricles, the one in the right cerebral hemisphere and the lateral ventricle in the left cerebral hemisphere, will drain into a collective structure called the third ventricle. And the third ventricle is found essentially within the diencephalon. It's found between the two halves of the thalamus. It receives cerebrospinal fluid from the lateral ventricles, but also the third ventricle does contain some choroid plexuses that produce that cerebrospinal fluid as well, again by filtration. So here's the approximate location of the third ventricle. This is the kind of highlighted in a light blue here. This would be the hypothalamus. This would be the thalamus right here. So between the two halves of those thalamus structures, we would see the third ventricle and the choroid plexus that occupies it. So from the third ventricle, we have a small canal or channel called the cerebral aqueduct that passes between the midbrain and the pons. So that is, it passes through the diencephalon and into the brainstem. And the cerebral aqueduct connects the third ventricle to a structure that's located in the brainstem called the fourth ventricle. And the fourth ventricle we see is essentially located kind of near the pons and the medulla. And that fourth ventricle has little openings in it called apertures, and those apertures allow the cerebrospinal fluid to drain into the subarachnoid space. So here from the third ventricle, we see the cerebral aqueduct passing from the diencephalon into the brainstem here, and so that cerebral aqueduct will eventually end up in the fourth ventricle. So cerebrospinal fluid that's accumulating within the lateral ventricles, and the third ventricle will drain into the fourth ventricle. The fourth ventricle has these small apertures within it that are allowing that cerebrospinal fluid to leave the ventricles and enter the subarachnoid space, which is shown here kind of surrounding the outermost layers of the brain. The fourth ventricle also contains a choroid plexus, which is producing cerebrospinal fluid. From there, from the fourth ventricle, that fourth ventricle will actually drain into a small channel within the spinal cord, kind of deep within the spinal cord. That's called the central canal. And the central canal will also carry cerebrospinal fluid to kind of the most inferior parts of the spinal cord.